fascinating world of DNA replication. DNA, or deoxyribonucleic acid, is like the instruction manual for building every part of a living organism. Its double helix structure is made of 2 strands, held together by nitrogenous base pairs, and there are 4 such types of pairs found in DNA: adenine, thymine, guanine, and cytosine. Adenine only pairs with thymine, and guanine only pairs with cytosine, and vice versa. The bases are held together with hydrogen bonds. DNA molecules also consist of deoxyribose sugar and phosphate groups. Before a cell can divide, it must create an exact copy of its DNA. This ensures that each new cell gets a complete set of instructions. But how does this incredible process happen? Well, that's what we'll explore in today's DNA replication quest. In order to demonstrate the process of DNA replication, I have considered an easy-to-understand gaminology of the process called the DNA replication quest. To complete our quest, we need help from some special molecular tools. Meet the team. This is helicase, the unzipping enzyme. Its job is to unzip the DNA strands by breaking the hydrogen bonds between base pairs. Next, we have primase, the starting point specialist. It lays down RNA primers to signal where DNA copying should begin. Here's the star player, DNA polymerase. It's responsible for adding complementary bases to build the new DNA strands. Finally, there's ligase, the fixer. It seals any gaps in the DNA, especially on the lagging strand. More on that later. Let the quest begin. Here's how DNA replication works, step by step. First, helicase enters the scene. It unzips the double-stranded DNA, separating it into 2 single strands. This creates what we call the replication fork. In order for the strands to stay separated, small proteins called single-stranded binding proteins bind to each of the strands, preventing them from joining back together. Next, primase comes in and lays down short RNA primers. These primers act like guideposts, telling the next enzyme where to start building. Before heading forward, it is important that I explain the concepts of leading and lagging strands. The deoxyribose sugar groups in DNA are actually made of carbon atoms. Therefore, we usually call it the 5-carbon sugar. If we look at the way it's structured here, the top or start of the strand contains the 5th carbon of the deoxyribose sugar, and the end or bottom of the strand contains the 3rd atom of the sugar group. Similarly, for the other strand, it runs in opposite ways. Hence, we can say that the first strand runs from 5' to 3', while the second runs from 3' to 5'. This is important to note, since DNA polymerase builds the new bases in the 5' to 3' direction. It becomes difficult to build the other strand because as helicase keeps unwinding the strand, DNA polymerase has to run all the way up to the new unwinded point, even if it's not finished with building the earlier bases. This strand would be called the lagging strand, while the other is called the leading strand. Now let's continue. After primase finishes its job, DNA polymerase takes over. Its job is to add complementary bases to each of the strands in order to exactly replicate the DNA. On the lagging strand, DNA polymerase works in small chunks, creating fragments called Okazaki fragments. Finally, ligase comes to the rescue. It seals the gaps between the Okazaki fragments on the lagging strand, completing the process. And just like that, our DNA replication quest is complete. 2 identical DNA molecules are created from the original, each ready to guide the instructions for a new cell. Remember, this process is vital for growth, repair, and reproduction in all living organisms. Thanks for joining me on this adventure, and now you're a DNA replication expert.
Can I be your star tonight? You fill